My name is Thomas Funck, and it's with great pleasure and great honor that I accept the Anne and Richard Sievers Award. I wanted to extend, first of all, my thanks to the Sievers family. This is really a privilege and a great opportunity, and I'm really excited to present my research with you today. So the overall topic of my PhD has been trying to quantify neurotransmitter receptor distribution in living humans. So I'm going to start this first by giving a bit of background overview over in vivo brain imaging, especially with respect to neurotransmitter receptors. So I don't think it'll take much convincing to tell you that neurotransmitters and their receptors are extremely important. They underpin all information processing in the brain because they mediate electrical signal conductance from one neuron to another. They're also the major pharmacological targets for many psychiatric conditions such as depression, anxiety, and schizophrenia. But how do we actually study neurotransmitter receptors in vivo, that is, in living human beings? So the main brain imaging modality to do this, and in fact really the only one that can do this, is positron emission tomography, or PET for short. And so PET is really cool because it lets you quantify neurotransmitter receptor distributions in living subjects. The downside, however, is that the resolution of PET is not great. Here at the MNI, we're very fortunate. We have one of the best PET scanners in the world, but it only has a resolution of up to 2.4 millimeters, which is pretty good, but is not as good as we might like. So the overarching sort of question or theme behind my research has been, how accurately does PET measure neuroreceptor density? And so the first part of my PhD was more of an empirical investigation into this question, in particular in the context of ischemic stroke. And stroke is a really interesting sort of challenging test case for looking at PET resolution. And this is because the damage that's caused by stroke is heterogeneous, it's very complex, and it's also highly localized to one specific region. And so hopefully the insights we get about PET resolution from studying stroke will then apply to other pathologies such as Alzheimer's or other types of dementia. Now, I want to convince you first that accurate quantification is very important. And to do that, I have a sort of a toy example here to look at. So you have two images, and you might wonder, do these two images come from the same brain? Just looking at these, you might think so. But in fact, if we look at the images that generated these two observed images, we see that they're in fact not the same. The image below here has thinner cortex, but with more activity. So the increased activity in the cortex here sort of offsets the thinness, and so you end up with two images that look very similar. Now, from a research perspective, or even a clinical perspective, this is very important. We have to be able to distinguish between these two conditions, and that's where PET resolution comes into play, because we need to be able to distinguish between, for example, cortical thinning or neuronal loss. And so the question that I've sort of been addressing in the context of stroke is, can we use PET to measure small changes in neuroreceptor density after stroke? And as a sort of corollary to that, do resolution enhancement algorithms actually improve the accuracy of PET? Do they make a difference in practice? Ischemic stroke occurs, as you may know, due to a blockage in the arteries. This blockage leads to oxygen deprivation and eventually cell death for a particular localized region of the brain. And this creates, of course, a region of dead tissue called an infarct. The infarct is not particularly clinically or interesting or it's not interesting in a research context either, because it's dead and you can't do anything with it. You can't really salvage it. There's not much to do there. However, around the infarct, there is a region called the Periinfarct cortex. And this region is interesting because it's the site of sort of long-term changes that can happen from the hours, days to weeks after the stroke. So these can include, for example, persistent inflammation, changes in gene expression, angiogenesis, axon spreading, remapping, and so on and so forth. So this is quite interesting because it's a locus for potential clinical intervention. Whereas the infarct is dead and useless, the periinfarct maybe can be saved, maybe the remapping can be enhanced, and so it's really interesting to study this region. But how do we define the periinfarct cortex? So traditionally, measures or methods to describe it have used a simple algorithm where you just draw a straight line from the border of the infarct outwards. Now this has the advantage of being simple, but it's problematic because it doesn't actually correspond to the brain's anatomy. Because the thing is, the brain has a very complex folded topology, and so if you imagine that these are two sulci and you want to measure the distance from the top of one sulci to another, if you measure it as a straight line, you're actually going to drastically underestimate the real distance that it takes across the cortical surface to go from one point to another. So measuring distances in the brain as Euclidean distance is not a very good idea, especially in the context of stroke for defining the periinfarct cortex. As a sort of improvement on this method, what I proposed was that first we define the stroke lesion on the MRI, as is done traditionally, but then we project the lesion onto a cortical surface representation of the brain. From there, I designed an algorithm that calculates distances across the surface, and essentially calculates the geodesic distance, as it's called, across the surface of the brain. What this lets you do then is create these concentric rings, in this case of 3 millimeters, around the infarct. And these are used to then study the periinfarct cortex using PET, and therefore studying the periinfarct cortex, studying changes in neurotransmitter receptor density in the periinfarct cortex. Now, the type of PET that we used in this particular case, in this particular study, is flumazenil PET, or rather the radiotracer is called flumazenil. And flumazenil is interesting because it binds to the alpha subunit of the GABA A receptor, which is located primarily on GABAergic interneurons. And this is useful because it's sort of a proxy measure for neuronal density, because these interneurons are more or less evenly and uniformly distributed through the brain. So by using flumazenil, we essentially get a proxy measure for neuronal density. So the data, we had 11 subjects that were scanned 2 weeks and 6 months post-stroke, and for each subject we acquired a flumazenil PET scan and MRI. Now, the results from the study are as follows. On the left plot here, you see the results from the initial scans. That's 2 weeks after the stroke. Here are the results from the follow-up scans 6 months after the stroke. On the x-axis here, you have the distance, so the periinfarct ring, the distance of the periinfarct ring from the border of the infarct. So if you go from 0 to 3 out to 15 millimeters. On the y-axis, there's a measure called BPND. This can basically be roughly thought as a measure of the density of GABA receptors. And so what do we see? So the dotted line here is the GABA receptor density in the unaffected part of the brain, the hemisphere that has not been affected by the stroke. The solid line, however, represents the GABA receptor density in the affected part of the stroke. And so what we see is when we get closer to the infarct, there's a bigger difference between the GABA receptor density in the healthy tissue in the unaffected hemisphere versus the GABA receptor density in the affected hemisphere. So we see that there's a periinfarct cortex region of about 3 to 6 millimeters where you have decreased neuronal density as a proxy, or which is measured as, or for which GABA receptor density is a proxy measure. Now, when we go to the follow-up study, what we see is we actually have an increase in the difference between the unaffected region and the affected region. So what this means is that we have decreased GABA receptor binding in the affected hemisphere, and this correlates to a decrease in neuronal density. So essentially what we have is long-term neuronal loss 6 months after the stroke. Now this is significant because normally when we think of stroke, we think that the primary damage happens within the first few hours. But what this suggests is that there's actually potential long-term pathological conditions that continue, and these could potentially be treated. So for example, we hypothesize that this long-term damage may be due to persistent neuroinflammation. So if we had some sort of treatment for that, we could potentially improve outcomes for patients. Now, I briefly mentioned PET resolution enhancement before, and this was the second part of this study. So in my master's degree, I developed an algorithm that can improve the accuracy and spatial resolution of PET, and we applied it in this context because we wanted to see if it made a difference to the GABA receptor densities that we measured. So this is just an example of what an image might look like before enhancement and then after. And what we found was that using partial volume correction, that is resolution enhancement, we could actually find significantly different results than when we didn't use the partial volume correction. And what this suggests is that using resolution enhancement genuinely improves the resolution and leads to more sensitive detection of long-term neuronal cell loss. But this leads to, of course, many other questions. For one, how do the changes in neuronal density relate to clinical outcomes? This is something that we're still looking into. And within the context of my PhD, the three following questions were especially pertinent. One, what is the maximum resolution at which we can observe changes in the periinfarct cortex? Could we have looked at changes at the 1 millimeter scale to get a more fine-tuned description of what was going on in the periinfarct cortex? How accurately are we actually measuring GABA A receptor density with flumazenil PET? Are our error levels on the scale of 2 to 3% or something more like 10 to 15%? And finally, how much does resolution enhancement actually improve the accuracy and the spatial resolution? So these are all things which are as yet unknown and which I hope to address in the second part of my PhD project. So switching from a more empirical investigation to a more theoretical one, I was hoping to answer the questions that I just raised with PET simulation. So I'll explain what that means. So in a typical PET scan setting, you have a patient, you put them in the scanner, and then you get some sort of observed image. The thing is, what we're not quite clear about is exactly how does the observed image relate to the brain that was actually in the scanner. And this is what I want to investigate. Now, if we knew the ground truth of the radiotracer distribution, or the neuroreceptor distribution of the brain we put into the PET scanner, then we could do a direct comparison between that brain and the observed image. But that's a difficult thing to do. So instead, what we can do is we can take a digital image that represents neurotransmitter receptor distribution, put it into a program that does PET simulation, and then get a realistic simulated PET image out of that. So fortunately, there are several PET simulators that exist and that are freely available. The one that I'm using is called GATE. Now, GATE is very cool because it uses software that was developed at the CERN to do particle physics simulation. So it's top-notch in terms of modeling actual physical processes. And so some very clever people took this software from the CERN and then created a sort of wrapper around it to do simulation for nuclear imaging. So we have the PET simulator. What about the digital neuroreceptor image? How do we get that? And that leads to the final part of my PhD, which is creating an ultra-high resolution neurotransmitter receptor atlas. So as some of you may know, a few years back, my lab, MSIN, of which my co-supervisor, Alan Evans, is the PI, along with collaborators at Jülich in Germany, published The Big Brain. And The Big Brain is a 3D reconstruction of 7,500 histologically stained sections from a human brain. And it's very cool because it's the first time we have an ultra-high resolution anatomic atlas of the brain from a single subject. And it's important to keep in mind what the Big Brain shows us is cellular density. What I'm proposing to do, or what I have started doing rather, in the context of my PhD, is something similar to the Big Brain, but for neurotransmitter receptors. So again, with collaborators from Jülich who have actually done the difficult wet work of taking brains, slicing them up, sectioning them, staining them, and then scanning them, we have this new data set that they have provided for us. And it consists of not one, but 3 human brains. And each brain is scanned sequentially for a different neuroreceptor. So there's 19 neuroreceptors in total that repeat sequentially. So the first batch of 19 slices will be one set of neuroreceptors, then the second batch will be another set. So you start off maybe with dopamine, serotonin, GABA, and then you keep doing all of the neuroreceptors, and you start over dopamine, serotonin, GABA, and so on and so forth. So what these images show you then is an example from each one of these 19 different neuroreceptors. And so what we want to do again is to take these 2-dimensional slices of neurotransmitter receptor density and then put them back into 3D. So this is just what I mentioned. We want to reconstruct the neurotransmitter receptors into some sort of version of the big brain, but for neurotransmitter receptors. And then ultimately, I want to use this to do realistic PET simulation with an absolute ground truth of neurotransmitter receptor distribution. Of course, this is easier said than done. because there's a lot of problems or difficulties with these two-dimensional slices. One is that the different neurotransmitter receptors have different intensity profiles, as you can see here. So it's not clear that it's going to be easy to match one onto the other to align them into a full 3D volume. Another problem is that the brains were not fixed when they were acquired, because they had to be fresh. And so this leads to all sorts of flattening out, to deformations and such that have to be corrected in some fashion in order to have a realistic looking image. Another problem is that we're just missing a lot of the slices. So we only have about 2,000 slices out of a total of 7,000, so that leads to problems. And many of the slices are incomplete and partial. There's also challenges related to the way the slices were acquired. So you can see that some of them have these sort of bounding boxes around them. Some of these have arrows. Some of them have smudging. Some of them have this white background. And all of this makes it really difficult to create some sort of automatic method that is going to allow you to align them one to another. So I created a reconstruction pipeline to do this as automatically as possible, because of course there's a lot of slices, you don't want to do this manually. And so I start with the raw images. And the first step is to remove the bounding boxes around the brain slices. And the way I did this is that I trained a convolutional neural network to automatically identify the bounding boxes and then remove them from the images. So that's step one. Step two is to do some sort of automatic cropping, because if we're only interested in this tissue, we have to get rid of the other pieces of brain tissue. And so I came up with a method that uses sort of standard image processing tools, but actually works quite well to remove extraneous brain tissue from the slices. You can see it works quite well here, where the target tissue we want is here, and the algorithm correctly removes all of the other pieces of tissue, and we're left only with the one that we really want. Okay. And so now we get to the tricky part of aligning these neuroreceptor slices to one another. And that, again, that's difficult because they have different intensity profiles, so it wasn't obvious from the beginning that this would work. So I'm going to show you an example, a couple of examples of the co-registration between one slice and another. So if you imagine that this slice here is fixed in place and this one is moving, we're trying to map this one onto this one. And so there are standard algorithms to do this, and it turns out that they actually work quite well for this particular use case. So here we see the overlap between the two images after the alignment, and it's quite good. We see that the co-registration technique is robust even when the slices look very different from one another. So again, this slice is fixed in place, and this one is being mapped onto this one. So we see after the co-registration, after the alignment, the overlap between the two is very good. Same thing here, good overlap between the two. Doesn't always work though, so I want to show an example where it's not perfect. In fact, it's quite bad. So here again, we're trying to map this one onto this one, but the alignment just didn't work in this particular case. However, overall, it does work quite well. And it's allowed us to reconstruct a full hemisphere for one of the human brains that we have. So it's not perfect yet because we still have to correct for the deformations from the fact that it's not fixed, but we've made a lot of progress so far. So here we are going through the brain sagittally, and here we're going through it axially. Here you can see the caudate nucleus quite briefly. And so I'll let the GIF run for a little bit. You can see that there are sort of like slabs that are well aligned, but then the slabs are not aligned to one another. So that's still an ongoing problem that we're solving. Okay, so the next step is now that we have an initial alignment of the receptor slices and we have them in 3D, we have to find some way to bring them back onto the subject's MRI, because the MRI was taken before the brain was removed from the skull, so it still has all of the, its anatomy is still intact, it hasn't been deformed. So what we need to do is figure out how to map the receptor volume onto the MRI. And to do that, I'm working with neuroanatomists, in particular Nicola Palomero-Gallagher at ULIC, who is identifying tag points that correspond between the MRI and the receptor volume so that we can eventually align these two together. Once we've done that, we will finally be able to correct for the nonlinear deformations that come from the fact that the brains are not fixed by using nonlinear co-registration with the MRI. So that's the last step. Hopefully we'll get to that soon, and we'll be able to launch the simulations. So there's still a lot of outstanding problems, and thanks to the support of the Sievers family, we're going to be able to continue making progress on this and solve the outstanding problems. Specifically, there's a lot of gaps, like I mentioned before, between the slices, and we have to find some way to fill in those gaps in some manner. Another problem is that we don't necessarily want one volume with 19 different neuroreceptor slices in it. That's not super useful. What we want actually is 19 different volumes, each for a different neuroreceptor, and so we have to figure out how to do that. Again, as I mentioned before, there's the deformations due to the fact that the brains are unfixed. That's still an outstanding problem. And so the future work that I hope to continue doing with this is not only reconstruct one brain, but all three of our human brains. There are also 3 macaque monkey brains, which will be very interesting, and many rat brains that can be reconstructed. So again, these are all neurotransmitter receptor slices that need to be reconstructed into 3D. And what this will afford us the opportunity to do is evolutionary comparative neuroanatomy in fully 3-dimensional space. So that's very cool. So finally, to kind of summarize, what I still aim to do before the end of my PhD is take these neurotransmitter receptors. Oh, the GIF isn't working, but that's okay. So this is supposed to be the neurotransmitter receptors here, and put them into, oh, okay, there it is. It's coming slowly. Take these, put it into gate, and then eventually come up with a realistic simulated PET image, which is based on a super realistic PET simulator and an absolute near ground truth representation of neurotransmitter receptor distributions in the human brain. Then we will finally be able to compare how well a realistic PET image, a realistic simulated PET image compares to what we put into the PET scanner. And we'll be able to really assess how well PET is working to quantify neuroreceptor densities. So I'd like to acknowledge the many people that have helped me along the way. There's, of course, my supervisor, Alexander Thiel at the Jewish General, my other supervisor, Alan Evans at MSIN here at the MNI. Also, I'd like to thank Claude Lepage and Paul Jean Toussaint, who have been my mentors within MSIN and who have provided countless hours of help and advice and support, and without them I definitely would not be here today. Of course, I'd like to thank our collaborators at ULIC, Carl Zillis and Nicola Palomero Gallagher and the rest of their lab, because they've collected the raw data that I'm working with, and they continue to provide help and advice. And of course, I'd like to thank again the Sievers family for this incredible award. This is such an opportunity, and I'm really thankful. Thank you very much.